Hey guys, just wanted to give you a quick, hopefully maybe less than 20-minute review over AP Bio topics, units 1 through 6 that'll be covered on this year's AP Bio exam coming up on Monday. So try to get at it immediately. Unit 1, we basically talked about biochemistry. There's also atoms where you have protons, neutrons, electrons, but we mainly focus on properties of water. Water being a polar molecule, meaning that it has a partial positive and a partial negative side to it. Oxygen being very highly electronegative, very corrosive. It is going to be formed by hydrogen bonds, and that polarity is going to dictate how they form with each other. That's also going to be responsible for trapping air inside of ice, which makes it less dense and float on water. It is the universal solvent. It dissolves most things, which allows our bodies to take up nutrients and various other medications. Cohesion would be another property that was typically water sticking to water. And then you also had adhesion, water sticking to other molecules. And the adhesion, you can see here, is responsible for capillary action. It also plays a role in transpiration in plants. Water does have a high specific heat. Due to this cohesion, water is not going to really change its temperature very much. That's why you can have a swimming pool sit out on 100-degree days for multiple days, and the water never actually warms up to 100 degrees. Also, our bodies, we don't get 100 degrees when we're outside on hot days. Heat of vaporization would be another one. That's going to be due to the breaking and reforming of these hydrogen bonds. Whenever the water evaporates from our skin, it actually helps cool us down. So sweating is a good example, panting and other animals. And it also helps moderate temperatures on this planet, the oceans being a very good example of that. Moving on, we had organic macromolecules. We have carbon, carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates, for the most part, are always going to be carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio, glucose being a good example that we use in class. Notice that there's no nitrogen or phosphorus in there, so if you get questions that pertain to nitrogen and phosphorus, it is not pertaining to a carbohydrate. They're typically used for energy storage. It's going to be glycogen in animal cells. You're going to have starch in plant cells. It can be used for structure, whether it's cellulose in plants, chitin in these insects and fungi, peptidoglycan in certain bacteria. And then we also, it's a good part to mention hydrolysis, dehydration reactions. If you remember, we're going to have to dehydration to link these various monomers, and then you also have hydrolysis, which is adding the water to break down various polymers. If you remember in class, we talked about we're going to need water in order to break down the hamburger that I'm eating. Lipids would be the second macromolecule that we talked about. They are hydrophobic. They typically exist in two forms, saturated and unsaturated. Saturated is going to have nothing but hydrogen bonds to every carbon, so it is going to be solid at room temperature. Unsaturated fats are going to be liquid at room temperature. They do have a carbon-carbon double bond, which creates this kink in the tail. And that's going to actually help maintain fluidity within the cell membrane, because if you remember, as soon as the cell membrane stops, then you start to not have transport across it anymore. It also is going to help prevent these plant cells from freezing when it gets really cold, because it creates a little bit of space in between the molecules. The next one that we talked about is proteins. Proteins consist of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Pay attention, the nitrogen is a dead giveaway that you're usually talking about a protein. Amino acids are going to be the monomers of that. There are 20 different amino acids that exist. Remember that the 3D shape is the overall goal of these proteins. That's going to determine their function, and there's 4 levels of protein structure. Because a mutation in any one of those levels can denature this protein and make it non-functional. So the primary level is going to be the amino acid sequence. The amino acid sequence sometimes plays a role on test questions alongside a DNA question or transcription or translation. The alpha helix and the beta plated sheets, that's going to be your secondary protein structure. That's typically due to the hydrogen bonds. Then you have tertiary structure, which is going to be due to various bonds, covalent bonds, hydrogen bonds, van der Waal interactions. You also have disulfide bridges that are occurring, and that's going to create this 3D shape of the protein itself. Now, quaternary structure applies to some proteins, not all. Hemoglobin is the example that we use in class. Hemoglobin has 4 subunits, therefore it carries 4 oxygen molecules. If you have a mutation or a disruption in this, then you could lead to, let's say, sickle cell anemia, where you only have 2 oxygen molecules being carried, and that's going to lead to complications down the road. Proteins is a very important macromolecule that we talk about in class. Enzymes being a very good example of that. Enzymes are going to be proteins that function as biological catalysts. They're going to increase the rate of the reaction by lowering the energy of activation. They're not altered or changed by the reaction, and they don't actually cause this reaction to occur. Keep that in mind. All enzymes have optimal conditions. That is a very good test question. If you change the pH, the temperature, the concentration, you may denature this enzyme and it doesn't even work anymore. The active site of enzymes is where all the action's going on. If that substrate cannot fit within the active site, then the enzyme's not going to function, and you could have all kinds of issues from there. Common enzymes that we talk about in class: lactase, DNA, RNA polymerase. We have ligase, helicase. That's all in DNA. You have proteases. We even have restriction enzymes that we talk about when it comes to DNA technology. When it comes to the types of reactions, they're going to be broken down between catabolic and anabolic. Catabolic is the breaking down of a molecule that's releasing energy. Cellular respiration is a very good example of that. And the Gibbs free energy constant is going to be, or the Gibbs free energy value is going to be a negative value because you're releasing the energy and now you're at a lower energy state in your products. The opposite of that, anabolic, that's going to be photosynthesis. You're building up this energy-rich sugar molecule that has a lot of energy to it, so it's going to have a positive value to it. The last one that we talked about is nucleic acids. That's typically going to be DNA or RNA. DNA being the genetic material, and RNA typically is going to be an intermediary between the DNA and the actual protein. That's going to be mRNA. You do have rRNA, which makes up ribosomes. Then you have transfer RNA, which is going to be playing a role in translation. They typically are going to be consisted of a sugar and a phosphate backbone. Remember that the sugars are different between the DNA and RNA. They are going to consist of purines and pyrimidines. That's where you have all your bases. DNA is going to have adenine, thymine, cytosine, guanine. RNA is going to be adenine, uracil, cytosine, guanine. So pay attention to that. Also, RNA is single-stranded. Remember, it has ribose for the sugar, things like that. We have phosphodiester linkages that are going to be occurring. That's going to be making these large polymers when it comes to nucleic acids. The next unit that we talked about is cells. Cells are the main focus of our class, I feel like. That's going to be all cells are going to be wanting to maximize their surface area to volume ratio. They want to have a very large surface area to volume ratio. So the smaller the cell is, typically, the larger the value for surface area to volume it'll be. Seems to be the key of life. If you notice, surface area is going to help plants, whether it's in the root hairs of those plants, alveoli in our lungs, villi in the intestines. This helps with absorption. If you remember mitochondria, chloroplast, the electron transport chain needs to have a lot of area in order to be able to carry out the process. Membranes would be another one, prokaryotic membranes. That's another way that surface area to volume is accomplished in prokaryotes. Surface area to volume ratio is an equation on your formula sheet, so you do want to be familiar with that. You want to be able to get the surface area of a rectangle and then put that side by side with the volume of the rectangle as well. You could also do division. There are different microscopes that we utilize or we look at in class. You have the light microscope. You can look at living things on that. That's the benefit of that one. Scanning electron, you can see things very well as some of the images that I have here. Scanning is just 3D. You can see up to like 10,000 magnification, but they have to be dead because you're passing electrons through it. Transmission electron microscopes will be another one as well. When we move on, cells are typically going to be broken down into prokaryotic cells and eukaryotes. Prokaryotic cells are going to be bacteria. Eukaryotic cells are going to be pretty much everything else. Keep in mind a couple things, that all cells are going to have membranes, they're going to have ribosomes, genetic material, and cytoplasm. But it's mainly these eukaryotes that are going to have these complex membrane-bound organelles. Endosymbiotic theory applies to cells. That's typically going to apply to the energy-producing organelles, whether it's the mitochondria or the chloroplast. And it's the idea that this prokaryote basically engulfed another prokaryote, and instead of digesting it, it actually utilized it and started creating energy from it, and also was able to reproduce it. The endomembrane system in eukaryotes is extremely important as well. That's going to consist of the nuclear envelope, the smooth and the rough ER, the Golgi apparatus, lysosomes, vacuole, and they're also going to have vesicles within there, and that's going to allow for transport within a cell. Ultimately, it's going to be either the molecule could either be used by the cell itself or fused with the plasma membrane and go somewhere else. Be careful when it comes to the cell membrane. There's a lot going on with it. This diagram down here shows a lot of things. It shows your phospholipids. It shows your glycoproteins and various other proteins that are going to be attached to the cell membrane. You're going to have peripheral proteins. You're going to have receptors. You're going to have cholesterol in there. In animal cells, that cholesterol is going to help keep our membranes fluid as well. You also have transmembrane proteins and carbohydrate chains that are going to be used for recognition. That's something else to pay attention to. And a mutation in any of those could lead to obvious effects. It's not always bad. That's something that I do want to keep in mind. A mutation might end up being beneficial to the organism. Maybe it's going to prevent let's say, a pesticide from going inside of a plant or something. Things like that. Continuing on with cells, we have different types of transport. You have passive and active transport. Remember, passive transport is going to be from high to low. Your types of passive transport, diffusion, which is the movement of molecules from high to low. You have osmosis, and then facilitated diffusion, which is the movement of molecules with the help of a protein, typically. Active transport is going to be the exact opposite. That's going to be going from a low area to a higher area. So that's going to require energy. Endocytosis would be an example of that. Exocytosis, you could have your sodium-potassium pump. If you remember with active transport and passive transport, we had a lab where we measured the percent change in mass in potatoes, and then you tried to see if it was a positive or negative change. And then you're able to determine whether the solutions were hypertonic, hypotonic, or isotonic. Water potential is another one. That's an equation on your formula sheet. Try to remember that. You might want to go back and review some of the problems that we looked at. Keep in mind that the closer to zero that you are, you have a higher water potential. So typically it's going to go from the value of 0 to something that has a more negative number. So you might, in your response, have to say something like beaker to cell or from soil to the root or vice versa, things like that. Next unit we talked about is energetics, mainly talks about photosynthesis and respiration, the fun one. When it comes to photosynthesis, sunlight's going to be exciting these electrons, for the most part, in the electron transport chain. That is located, if you remember, in the thylakoids of the plant. That's going to be where light is going to excite the electrons. You're going to have water coming into that, and then you ultimately end up getting oxygen. You get a little bit of NADPH and ATP that's going to be used to power the Calvin cycle over in the stroma. But for the most part, that's where our oxygen is coming from. You have two different types, cyclic and non-cyclic, and that's mainly going to be determining what's formed at the end. Are we getting ATP or not? Photorespiration is something that plants sometimes will end up performing. Basically, Rubisco is going to pick up an oxygen instead of the carbon dioxide, so it's ultimately going to end up leading to very little sugar production, because if you notice the Calvin cycle, it is going to be dependent on carbon dioxide. There are different adaptations to this photorespiration. You have C4 plants, which are going to be utilizing two different types of cells, and they're going to keep them spatially isolated from each other. That way they carry out the processes separately. CAM plants would be another one. Cacti, a good example. They're going to open their stomata at night. This typically applies to plants living in hot temperatures. And they're going to be temporally separated between day and night. We also did another lab with this one. If you remember the photosynthetic rate of leaf chads, we kind of changed variables, whether it's the color of the light, the amount of light, or the type of plant, things like that. Respiration is the other one. Respiration is going to be the breakdown of food in the presence of oxygen. This is a catabolic reaction. If it's anaerobic, that means that you are going to enter the mitochondria. Oxygen will be the final electron acceptor in the electron transport chain, and you'll be generating the most amount of ATP. If you do not have oxygen, available to you, then that's going to be an anaerobic process. That's going to be occurring out in the cytoplasm for the most part. Lactic acid fermentation in animals, alcoholic fermentation in yeast is going to occur. And something to keep in mind between fermentation and respiration is that glycolysis precedes both events. Glycolysis is considered to be a universal energy producing process because all organisms have cytoplasm. Therefore, the very earliest prokaryotes could have produced their energy through glycolysis. This does utilize redox reactions. You guys also substrate level phosphorylation and oxidative phosphorylation, and that's the chemiosmosis that we talk about and spend some time on in class in the electron transport chain. If you notice the structure of the mitochondria, you're going to have the outer membrane, and then you got this inner membrane, and then you got this inner membrane space. And you're going to end up having protons that are going to be pumped to this inner membrane space, and then they're going to drive through ATP synthase, rotate that little protein, and generate ATP. That's the maximum way to produce ATP, it seems like. This proton gradient is going to be needed. So keep in mind that mutations or any kind of disruptions in this proton gradient is going to lead to a decreased amount of ATP formation, something to definitely pay attention to. In class, we did the fermentation, we did respiration rates in peas and goldfish. If you remember, goldfish are going to be cold-blooded, so their respiration rate is going to be affected by the temperature of the water. Next unit that we talked about is the cell cycle and cell communication. We basically talked about the cell cycle being mainly interphase, 90% of the time. A cell's just growing, replicating its DNA, and it's getting ready for mitosis. Mitosis is going to be where you actually see the first chromosomes. That's going to consist of prophase, metaphase, anaphase, telophase, and cytokinesis. If you also remember with mitosis, that could be a type of asexual reproduction in prokaryotes. You could end up having clones, and that could also be in plants as well. But it does only produce clones. Binary fission is in what's in bacteria. You could have growth, whether I need to grow from a baby to an adult, or repair. Let's say I cut myself and I need to generate new skin cells. Mitosis does go from a diploid cell to a diploid cell, something to keep in mind. And the difference between the cytokinesis in a plant and an animal. In an animal, we get the cleavage furrow. In plants, we end up getting this cell plate. Meiosis would be another one that's extremely important. That is a main source of genetic variation. That's going to be sexual reproduction. So you're involving gametes, your egg and your sperm. You're going to end up having that crossing over event. If you remember, that's going to create genetic diversity. You also have independent assortment, things like that. Segregation, diversity, evolution. Also keep in mind that meiosis is going from a diploid cell to a haploid cell. So you're reducing the amount of chromosomes because fertilization will eventually end up restoring that number. Mutations, cancer, non-disjunction, all those types of things are things that can occur in chromosomes and be detrimental. We also talked about the signal transduction pathway in Unit 4. The signal transduction pathway, really important. This is the basics of it. You have reception being the first step, and keep in mind that a mutation in this receptor protein could end up having very extreme effects. It doesn't necessarily have to be bad, but it could be that it prevents, let's say, a poison from entering the cell. So that might be a good example. But reception is going to be where a ligand is typically going to bind to a receptor protein. Then you're going to have transduction, the second step, which is going to be amplification of this signal. Cyclic AMP typically plays a major role in this one. Then you have your cellular response. And keep in mind that your cellular response could be anything. Maybe it's apoptosis. Maybe the cell needs to destroy itself. It could be the secretion of a certain molecule. It could be to activate transcription and maybe start making something, or growth. Maybe it just stimulates mitosis. You do have various types of cell communication. I threw in here autocrine, which is going to be self-signaling, so the cell's telling itself to grow more. That might be a tumor. Pancrean, that's going to be nearby cells. And then you have juxtacrine, which is going to be requiring direct contact between these cells. Immune system is the first thing I think of when it comes to that. Unit 5 is going to be heredity and genetics. If you remember in genetics, we had basically 3 types of dominance. Complete dominance, that's going to be where you have a dominant and a recessive allele. So black cat, white cat makes a all black cat. The heterozygote's black. In incomplete dominance, the heterozygotes are going to be a blend of the two parents. So black cat, white cat makes a gray. So the heterozygote is gray in color. It doesn't look like either parent. Codominance, you're going to have both alleles being expressed in the heterozygote. So you're going to end up having a black cat, white cat produces a black and white cat. And that does apply only to the heterozygote. We do have recombination frequencies. I do want to mention at this part that the higher recombination frequencies are going to exist for genes that are further apart on a chromosome. So if you see the image on the screen, the B and the VG, they have a 17% recombination frequency, which is higher than the other ones because they're far apart on the same chromosome. Now, if they're typically closer together on the same chromosome, then they're going to be inherited. They have a higher probability of being inherited together. Probabilities is something that I believe should be on your test. Remember that we had different genotypes, so you may end up having something that has 3 traits, let's say, and then they try to get you to come up with what's the probability of having the homodominant, the heterorecessive, and the homozygous recessive and the heterozygote. What's the chances of getting that out of those two parents? We also had the chi-square equation, the null hypothesis. If you remember, there's a couple things. You might want to go back and look at some of that work that we have done. Don't forget your critical value, your degrees freedom. A big hint I might tell you is try to remember what we should expect. Should we expect even distribution? Should we expect that there's 3 choices and there's 1/3 of a chance for each of them to go, for the organisms to go into each of them? So that'd be 1/3 or 33%. Also remember monohybrid crosses, that's typically going to have a 3-to-1 ratio, and then your dihybrid crosses, which have that 9-to-3 to 3-to-1 ratio. So those would be your expected values, and it helps you kind of figure out how many of the organisms should I expect to have this particular trait or perform a certain action. Another one that we talked about is pedigrees. Pedigrees are typically going to be showing the inheritance of a particular trait. They can exist as dominant, recessive, or they can be X-linked. Remember, X-linked traits typically are going to affect men because we only have one X chromosome. Most sex-linked traits are passed to the men from their mother because we inherit our X chromosome from mother. The Y chromosome obviously has to come from dad. Mitochondrial inheritance would be another one that is passed from mother to offspring. So if you see a pedigree and you only see it being passed on whenever the mother has it, that's probably a dead giveaway that it's a mitochondrial inheritance. If it's X-linked inheritance and you're looking at a pedigree, what you most likely will see is that only the men are going to be affected. And if it's recessive, it'll skip a generation. We also have mutations that play a role in this spot. Mutations might be point mutations, such as a substitution. Those can be bad, but typically not so much. Sickle cell anemia would be an example of a substitution mutation. Deletions and insertions are the bad ones that I want you to pay attention to because those are frame shifts, and those can end up altering all the amino acids down the line, and you have a protein that will not function at all. You can also have mutations in chromosomes as well. We also looked at karyotypes. If you remember on this one, we have Down syndrome on this one, and this looks like a female because there's 2 X chromosomes down here at the very bottom. When it comes to genetics, epigenetic inheritance is something else that is something that needs to be mentioned. That's going to be DNA methylation, histone acetylation, and there's going to be, it's basically the environment affecting your DNA. It's something other than the DNA code itself that is going to infect the inheritance and the passing on of various traits. The code's still the same, but maybe it's highly methylated. Maybe it's not. Maybe certain things in the environment that that person was exposed to is going to lead to them passing on or not passing on certain traits. Last unit that we talked about is gene regulation and expression. Start this one off by saying that the central dogma of biology, information always has to flow from DNA to RNA to a protein. It seems like it would be a lot more efficient if we could just go straight from DNA to the protein, but we can't. We have to make a copy of it because our DNA is locked up in the nucleus, and proteins are going to be made on ribosomes that are out there in the cytoplasm. DNA replication is going to be DNA making DNA. So if you'll notice, there's various enzymes involved: helicase, DNA polymerase, you have ligase. The big thing that I do want to want you to pay attention to is whenever you have a replication fork, your leading strand is always going to be facing or growing towards the replication fork or the origin of replication. And that is because DNA polymerase can add these nucleotides to the free 3' end as it normally can. Whenever it's working on the lagging strand, it's essentially going to be doing it in the opposite direction. So it's going away from the origin of replication, and DNA polymerase is essentially trying to work backwards. So it's going to be making it in small Okazaki fragments. Those fragments can be glued together later on by the enzyme ligase. Transcription being another process. Keep in mind, transcription is going to be occurring in the nucleus. That's going to involve your TATA box. You have your promoter area. This is going to involve RNA polymerase because we are now making an mRNA from DNA. It does involve 3 steps: initiation, elongation, and then termination. You also have translation. Translation is going to be out in the cytoplasm. For the most part, that's going to be utilizing that mRNA and a tRNA to assemble an actual polypeptide. Whenever it comes down to regulation down here at the bottom, I try to highlight green what's going to be promoting and what's red is going to be inhibiting. So you'll see here histone acetylation that can promote DNA replication. DNA methylation is going to inhibit it. Euchromatin is going to be where the DNA is actually loosely bound, so you can actually have all these enzymes get in there. When DNA exists as heterochromatin, for the most part, it's too tightly packed and nothing can get in there, so no transcription occurs. There is processing that occurs. Remember your intron removal, the addition of the 3' poly A tail, which is going to help degradation out in the cytoplasm. It's going to keep that mRNA stable while it's out in the cytoplasm. You also have this 5' cap, which is going to help that mRNA bind and locate the ribosome. Then you also have splicing, and that could be splicing, but is going to be where we're going to get multiple mRNA. So after all the processing, we have one mRNA molecule, but we can end up getting a bunch of different polypeptides from that one by cutting it in different spots. You also have ubiquitin, which is going to basically target a proteasome to destroy whatever polypeptide you created. Maybe it's just not needed. So those are all types of gene regulation, and they're extremely important. You definitely want to be familiar with those, whether it's gene regulation during post-transcriptionally or pre-transcriptionally, post-translational, pre-translational, things like that. Regulation, we basically talked about how in prokaryotes, they're going to have an operon system. They're going to have this operator area, and then they're going to end up having various other proteins, if you will, that are going to be needed in order to continue the process or stop the process. The first one's a lac operon. That's an inducible system. It's typically turned off, and it's also catabolic. So the one that we talk about in class is the lac operon. So if lactose does become available to these bacteria, they are going to want to have that lactose bind to the repressor. It's going to move, let the operator be free. The promoter can get in there and start carrying out transcription. That way they can make lactase in order to break down this lactose that they came across. The trip operon is going to be the exact opposite. That's going to be repressible. It's typically turned on, and it's also anabolic. So tryptophan being one of the amino acids that we mentioned in class. So would you want to make tryptophan if it's already available to you? Of course not. So this one is typically going to be turned on. Whenever tryptophan does become available, you have your repressor. It's going to bind there, and it's not going to, whenever the repressor is not bound there, it's not going to be, it's going to be making tryptophan. Excuse me. Sorry about that. Eukaryotes, it's a lot more complex. There's a lot of different things involved. When it comes to this, you have various transcription factors, enhancers, activators. You have the folding over of the DNA. You have all this stuff that basically makes it a little bit more complex in order to carry out transcription. Let's see here. When it comes back to this lac and trip operon system, I do want to just mention again, would you want to make lactase if you're not in the presence of lactose? Of course not. So you have your repressor typically bound there. When it comes to this tryptophan, if it is available, you do want your repressor to be there because you don't want to be wasting your time making this amino acid if it's already available to you. If it's not available to you, then the repressor will leave. Then you'll start making tryptophan. You'll have that wonderful amino acid to make whatever it is that you need. DNA manipulation is another one. We talked about DNA extraction, the spooling of DNA. You have gel electrophoresis, which is going to be the separation of DNA molecules. Keep in mind that DNA has a negative charge due to the phosphate groups in the heads. So it is going to be moving in the backbone, sorry. It's going to be moving towards the positive end because of that negative charge. The smaller molecules will travel a lot further, the heavier molecules, not so much. We also had a polymerase chain reaction, which is going to be essentially amplifying the amount of DNA that you have. So now you have a bunch of different copies in case you destroy it. You also have transformations and genetically modified organisms. And these things typically rely on these plasmids that are found in prokaryotes. Whether it's getting bacteria to make human growth hormone, insulin, various things like that, or getting plants to be able to have various nutrients within them, we use these plasmids. You also have restriction enzymes that could come into play, and they can end up cutting various, which is an example of a plasmid. They can come in and cut DNA at specific sequences. We talked about CRISPR, Cas9 in class. Hopefully the potential for that will expand and we'll be able to remove bad segments of DNA and hopefully replace it with good segments of DNA. We also did the transformation lab in class. If you remember, we got the piglo gene from the jellyfish and we inoculated it with E. coli, and then we put it on this various plates. These plates would either contain an antibiotic or would not. And if you remember the plasmid that we gave to glow, it also had antibiotic resistance on it as well. So if it grew on that plate, it was resistant to the antibiotic, and it was also glowing if it took the DNA from the jellyfish. On the plates where there was no plasmid giving, it would die, hopefully, or you wouldn't have any growth because it wasn't resistant to it didn't receive the plasmid from for the antibiotic resistance. A couple little random things I want to just mention to you guys before I finish this up is that graphing is going to be definitely a big part of it. You definitely want to be able to analyze graphs, and you also want to know parts of an experiment. So I figured this would be a good spot to go over positive, negative controls. Positive controls are going to show that your system can actually work, that your molecule is actually functioning. It has a known result. A negative control is going to show that the observed change is due to the variable that you're testing, not a placebo. It wasn't an accident. You're not expected to get any results whenever you have your negative control. You're just proving that your particular molecule or your particular variable in the experiment is what's responsible for the results. So I put a little example here. Say you want to test a new DNA polymerase. Well, if you have a new DNA polymerase, a negative control will be the experiment ran without the polymerase because you would not expect any results. A couple basics. You got your dependent variable. That is going to be the variable that's measured. That is on the y-axis. So that's going to be this vertical axis here. You have your independent variable, and that's what's typically changed by the scientist. That's going to be on the horizontal or the x-axis. So pay attention to that, what's on each axis. A couple things with genetic variation. You do have mutations, meiosis, sexual reproduction being a main factor in genetic variation, random fertilization as well. And at this point, I also wanted to mention in prokaryotes, transformation can occur, and that does increase genetic variation. That is using plasmids. Conjugation, that utilizes the pili, where the bacteria can actually just exchange genetic material directly with each other. And then you have transduction, which is typically going to be utilizing a virus. It's going to be going around spreading the DNA from bacteria to bacteria. But that's about it. I wanted to kind of keep this thing as quick as possible. I just wanted to give you something to kind of review, whether you look at it or listen to it. I want to wish you guys the best of luck, and I hope that was helpful.